Yeah. Oh, yeah. Sorry. I'm known. I will admit for a second. Let me give my welcome spiel. So, welcome to Mary Washington. Welcome to a mock lecture. This is a Destination Day event. I love Destination Days. They're actually my personal favorite as a faculty member. Yes, I am known for having unbelievable quantities of energy. I'm one of I would call myself the craziest faculty member in chemistry. So I am Dr. Randy Reef. I have been here now for 7 years, which is kind of crazy, actually. I no longer get to call myself the new guy. I used to be the new prof. But I'm an associate professor here in the Department of Chemistry and Physics. And my specialty is biochemistry and analytical chemistry. I've merged them together and call myself a bioanalytical chemist. I'm making up that title. But I teach classes from general chemistry to analytical chemistry to biochemistry. And one of the things that I loved was flow cytometry, which is what we're going to talk about today. But before I do, I want to note for everybody there that this meeting is being recorded. So feel free, if you don't want to appear on video, feel free to mute your microphone and turn off your camera. Although if you're willing to talk and respond, please do. We're going to do a little game here in a little bit, right? Where I would love the audience participation with the microphone, call out what you see. So feel free to talk. When I teach in class, you would all be in my room here, right? And I do a lot of question, wait for an answer, response. I'll try not to rely too much on that through Zoom. I know Zoom can get a little interesting for that, but it'll be fine. Feel free to talk to me. The other thing I will note, once we really get started, I'm kind of an old school prof. So if it were, I know it's weird, right? I've only been here 7 years, I'm old school, but I am. So my style would be to do basically this lesson on the whiteboard. And so I'm just going to warn you that in Zoom, the equivalent of that is a document camera. And so we're going to do some writing with markers and paper, right, with a document camera. But what that means for you as the audience member is that I won't really, once I switch to doing that, Zoom, I have trouble seeing the chat. So long story short, the chat won't be super reliable for me. So if there's something you can't see, or if you have a question, feel free to just interrupt me and be like, Dr. Reeve, what about this? Feel free to do that. And my students do that all the time, right? And that's how I teach. So feel free to do that. The chat box, I won't be able to see until later. And so after we get done with the mock lecture portion of today, I'm saving some time basically for a Q&A. And so you'll be able to write questions in the chat box or ask us questions. And so I will be here and I will totally give you basically my perspective as a faculty member on any questions you may have, whether that's about the biology curriculum, the chemistry curriculum, anything like that, how science classes work. I'm here to do that. I will answer questions when we get done. Okay, so with that, I guess we will get started now. So I know that this slide I've got sharing, have no fear, it's shared up on the screen. It's the only slide you'll see. Again, old school professor. And it looks a little bit intimidating, right? But today we're going to talk about how we analyze blood, right? And so let's imagine, for instance, that you go to the doctor, right? You go to the doctor's office and the doctor, thinks, well, you might be sick, you might be fighting some kind of infection, bacterial or viral. And so they take a blood sample, right? And so then they call you, hours later, right? And they're like, oh, well, your white blood cell count is elevated. So, we're going to put you on this antibiotic, right? And so the question would be, well, how do they do that? How can they do that in, say, hours, right? They get my blood sample, they send it off to a lab, and what happens to it? And that's what we're going to talk about today. And what happens to it, to answer the question, is basically your scientists, right? Your medical scientists will basically your medical techs will run a flow cytometry analysis of your blood. And so they'll put it through this big instrument we've got there in the center, right? That looks very intimidating. It's a big box. But when you simplify it down, it's really not. It's actually really cool. And so the job of a flow cytometer is to analyze cells. And what do I mean by analyzing cells? Well, basically what it's going to do is it's going to it'll measure things like the size of a cell. We'll talk about how. It can measure things like the shape somewhat. Shape is a little bit fuzzy a term, but for simplification, we'll call it shape. And then we can also do things like analyze color using special probes that make certain these molecules that we incubate with the cells that'll make certain cells glow certain colors. And so it can tell basically the person reading the data whether you've got lots of T cells, like the T lymphocytes there on the bottom of that bottom right corner, or like B lymphocytes. And so we can count those cells. We can see here that we have this large density of T cells, 66% is what it's saying there. And so we get data like that. And so we're going to walk it through. Have no fear. We'll walk through how it does this, and we'll keep it simple. But what we're going to do first is actually we're going to do a demo. And so I haven't ever, this is the first time, bear with me, this is the first time I've done this on Zoom. So this is going to be pretty exciting. But I'm actually going to stop my share. And so that way I can see everybody, right? And, oh, well, I could see everybody until my computer decided to do crazy things. Come back, Zoom. Okay. So now what we're going to try is we are going to be the flow cytometer, right? So we are going to pretend that we're the flow cytometer, and we're going to analyze some plastic balls. Are you ready? So basically what I'm going to do is I'm going to drop these cells down. I'm going to stand up and I'm going to drop these cells, they're balls. And I want you to tell me what you see. You see green balls, green cells, you see blue cells, big cells, little cells. We're going to give this a shot. Are you ready? I know I have it camera down because that's, I want you to be able to see the balls. All right, ready? Here we go. I'm going to try not to send them all over the room. What did you see?
Orange, blue, green.
Yeah, right. Yeah. You saw like orange, blue, green. It was really chaotic. Here, let's try it again. Let's try it again. Right? And tell me how well this works. You ready? Okay.
Purple.
Purple. Oh, we got purple now. Okay, so if I just took the blood sample, if I just took the vial of blood, and I basically said, look at all the cells. It'd be like, wait, how? How do we do? What do you mean look at all the cells at once? That's crazy, right? How am I supposed to get anything meaningful from all these cells, all being there at the same time? But what? What if we were able to funnel the cells so that the cells drop down in front of you one at a time? Now, it's still going to be fast. It's still going to be pretty challenging because they're going to flow past you, right? But we're going to try this out. Are you ready? I'm going to drop a cell. I'm going to drop a cell, right? And you're going to tell me what you can figure out about it. Are you ready? Here goes the first one.
All purple. Yeah.
Purple. Now, compare this one.
Big blue.
Big blue. Huge. We just got it. Okay, here we go. We're going to go do another one. Ready?
Yellow.
Yellow. How big relative to the other one?
Pretty small.
Pretty small, right?
Orange.
Orange. This is so cool. Flow cytometers do exactly this. So the flow cytometer system, right, which is what we're going to get into now. Now we're going to switch. Demo's done. So now I'm going to try switching us into lesson mode. Ready? Where I'm going to write on pieces of paper. It's going to be hilarious. Here we go. Let's talk about flow cytometry. Let's break down how they do this. What if we use the funnel, right? So if we use a funnel, basically, if we can create some kind of funnel, oh man, how do I look? Did we lose me? My camera just shifted. Can we see me at all?
We can still see you.
Okay, good. Okay, my camera, hey, focus. That's the downside of it. So here we go. I think it'll help if I write something. All right, so ready, we're talking flow cytometry. Here we go. Flow cytometry just means basically counting cells and analyzing them, right? So the idea is to count cells. So what the technique does is it counts cells and it analyzes aspects of them. And I know that's a broad word, right? And it analyzes them. Well, the really cool thing is what you just saw, what we were just doing. Come on, focus. Yeah, it's focused. What we were just doing would have taken forever, right? We just did one ball, like it was really slow, right? It took us what, like 30 seconds to do 3 cells. Flow cytometers can use the power of light. They use light and optics and electronics to analyze up to 10,000 cells per second. But they do fundamentally the exact same thing we just did, right? So they're going to look at cells, and they can look at size, and in some cases shape. We'll call it shape. I'm going to put shape in quotes. Shape is pretty non-technical. If you were talking to a biologist, they would argue with me about cell shape. They'd be like, that's not a great word, Dr. Reef. That's kind of true. They also will analyze number, because they're going to be able to count them. They'll know exactly how many cells we dropped, right, in front of the viewer, right? So we could count, well, we looked at 4 cells, right? We'd be like, what, 2 yellows, a purple, there's a yellow, a purple, a blue, and an orange, right? So we know we looked at 4 cells. And then what that does, the color aspect actually allows us to look at the type of cell. Now, we want to note that this is where it gets a little bit more complex in the science side, because we have to use labeling molecules to do that. And we'll talk very briefly about those labeling molecules, but that's not the focus of my talk today. So while I get very excited about them, it's not the focus. The idea would be that we can make certain cells in your blood a different color when the flow cytometer looks at it, right? Which is really cool, right? So we have to use extra molecules though. We have to use molecules that we incubate with your cells and let them bind for a bit, and they can make your cells glow. So the flow cytometer, the instrument is actually broken down. Am I losing my camera again? I feel like I'm losing my camera again. Is that better? The flow cytometer is actually broken down. I'm going to break it basically down into 2 big sections, right? So 2 big things we're going to look at. The first is the flow part of the flow cytometry, right? And what I'm talking about here really is the funnel. If you're thinking about it, basically in simple terms, we're talking about how we can funnel the cells to go one at a time. We can basically get them to be in line, and they can pass our viewer one at a time, right? And so how do we get cells in a vial of blood to do that? And so the answer is fluidics, right? Which is actually some serious physics that we're going to keep simple, right? Because physics scares me, right? I don't know about you, but it scares me. So what we're actually going to start out with is we're going to start out with the center, right? Center channel that's going to have inside of it our cells. It's got a bunch of cells, and so I'm going to put them as green marker here, right? We have this whole mess of cells, and you can see that they're clumped up. They're not in really a line here, right? So how are we going to get them into a line? Well, what we're going to do is we're going to put a kind of channel around them, and we're going to flow water or liquid. It's really not water, technically, it's a buffer, but we're going to flow fluid on the outside really fast. And so we're going to have a fast flow rate of liquid on the outside on either side of our central channel there, right? So this is fast flow, right? And what this is going to do is we're then going to draw kind of it actually does kind of look like a funnel, right? Right? What this is going to do by flowing the liquid really fast on the outside and flowing the liquid slow in the center, so the actual sample where your blood is, where your blood cells are, is going to be slow flow. What it actually does is a process called, big vocab word, don't worry about it, right? If you were in my class, you'd be worried about it. But don't worry, we're not going to take a quiz here, right? Oh, I lost my camera again. We're not going to take any quizzes here. It's okay. This is called hydrodynamic flow. So what's going to happen is the fast flow on the outside is going to actually push our cell liquid into a really small area. And so what this does is this now, where's my green marker? This now pushes our cells into a line. And so the cells now flow one by one in a line through the center of a tube. It's a very small tube, but the center of a tube. So we have effectively funneled our cells, right, into this tube. Now, how do we look at them? Well, we could look at them by eye with our little demonstration here. We could absolutely look at balls. We could drop a ball and we could see the different color and we could see the size, right? Well, the flow cytometer is going to do exactly that. The question is, how do we do that? And so it actually starts with one of my favorite things in the world, the laser. That's right, it's a laser beam. It's going to start with a laser. I love lasers, right? And this laser typically actually shines blue light, right? So what we're going to do is it shines blue light, and it's going to shine it straight at that cell there, right? And so when it hits this cell, that's right in front of it, although it should make the blue line connect for you so you can see that it hits, right? When the blue light hits this cell, the light is so intense that it's going to hit that cell and it's going to, first of all, it's going to bounce, right? So the light, the blue laser light, first thing it's gonna do is bounce off the cell. We call that scatter is the official word, right? And so you're gonna get some scatter of the blue light when it hits the cell. It's gonna disrupt it, right? And so the blue light's gonna scatter in multiple directions, right? The one direction is forward and one direction is to the side. Now, the side part, we're not going to worry too much about for now. And so this light.
Randy,
yeah?
we do have a question.
Oh, what's up?
So somebody wants to know, does the wavelength of the light affect it at all?
Absolutely. Okay, we're getting more complicated than I wanted to get for this audience. But yes, the reason it's blue is because blue light is actually used on purpose because the molecules that label it, they're designed, those molecules are designed to absorb the blue light and then glow other colors that are lower in energy than the blue light. So they're going to glow, as we're going to see when we get to the analyzer, in yellow, green, red. It's going to be awesome. Yes, the blue light is absolutely chosen strategically here, right? Now, the other reason blue light is chosen, and this is something that people don't really tell you very often, is because blue light, while it's actually high in energy, as you learn in your physics class or your chemistry class, the blue light is still visible. It's not going to hurt our cells. It's very important, right? So while it's high in energy, it gives us a lot of colors that we can look at on the other side of the Roy G. Biv spectrum without hurting our cell. That's the other reason we use blue, right? So yes, it does matter. Yeah, ask away.
Is that why we don't use purple light or UV light?
So, okay. So yes, fantastic question. Purple lasers are very hard to come by. It's actually very, very difficult to, one of the other reasons is practical in terms of the engineers. So engineers have had trouble making purple. They're better at using UV. UV is so high energy that just like UV cell damages your skin, you risk blowing up your cells. You'd be hitting them with a laser beam of UV light, your cell is going to die. It is going to like hit that beam and be like, pfft. Well, how am I supposed to analyze something that goes, pfft? Right? Not cool. So that's why they use blue. Other questions? I'm gonna ignore it.
You gonna ignore the other questions?
No. I was writing my next.
He's writing. He missed the question.
Yeah, what's up?
What are some factors of the blue light besides its wavelength that could affect the cytometry?
Honestly, the wavelength is the reason it's used. The factors would basically be the wavelength because the wavelength works really well with the labeling molecules. It works really well. The other factor is practical. It's inexpensive and easy to make a powerful blue laser. Now, you will notice though, in real life, that have you ever checked out laser pointers? Let me ask you how often you've seen a laser pointer that is blue. Have anyone seen laser pointers that are blue? What color are laser pointers usually?
Red, of course.
They're usually red, right? Because red is lower in energy. So red is much easier to make in a really small laser pointer device. Blue is actually really hard to do. This laser is still going to be big, by the way. It's physically going to be a box like this. You use argon ions, actually, is what they do to do it. This is going to be very high intensity blue light. There's a lot of blue light. Like you put your eye in it and your eye is gone. You will be blind if it hits your eye. Like it is still very intense in the instrument, right? So intensity matters, and we can make blue light very intense. And then the wavelength of the light will allow us to look at really cool colors later. Other questions? Don't worry, we're going to answer more. I'm going to talk about the colors. Are we ready to talk about the colors?
Now.
Okay. So the first thing that's going to happen is this light is actually that forward scatter, right, is going to get collected by a detector. And it's going to be really important for us later. And so we call this, by the way, when you see a flow cytometer, they'll call this the FSC, the forward scatter, right? The amount of forward scatter. Then there's actually a side here, but this view is not very good for drawing the side. So now what we're actually going to do, because we just transitioned from the funnel, we got the funnel, and now we started talking about the laser and the detectors. Now we're talking about the analyzer. So I'm going to switch modes here, and we're going to draw the analyzer. So the second part of a flow cytometer that we care about is called the analyzer. I'm calling it the analyzer. It would also be called the electronics and the optics if you wanted to get technical, right? I call it the analyzer. So now what we're actually going to do is we're going to take our previous image where we were looking at the sides, we were looking at the funnel, and instead we're going to flip our view to look down the funnel. So we're going to look down. I know it's really weird. This is going to be a top view. We're going to draw a pretty picture, I hope. I hope I draw a pretty picture. Don't tell me my picture's not pretty, by the way. It'll make me cry. We're going to go with a top-down image here, right? So I'm going to draw in kind of in the center of my page, so I got to build myself some serious room. I'm going to draw a box to represent our channel that we had originally, and I'm going to redraw our laser because our laser is actually still in the same place. So we've got our laser, we've got the box, and we've got our, as we already talked about, our first detector for that forward scatter, right? That FSC, right? And so now that I've got a top-down view, my cell is right here, right? So here's my cell. It would be, spherical, but I draw it as a circle. And I am shooting my blue laser, right? I'm sorry, shooting is the wrong word. It's irradiating. I'm irradiating that cell with my blue laser, all right? So my blue laser is flying in there, going to hit that cell. Some of that blue light is going to bounce off and is going to get collected by our forward scatter detector. Then, because I'm in a top-down view now, we can now look at another detector and look at the blue light that bounces off from the side, right? And so there's going to be blue light that bounces off from the side, and that's going to get collected by another detector, right? This is a detector I can't really write. I'm going to try and write in the, oh man, I'm outside. That looks sloppy, but all right. And this is what we call the side scatter detector. This detector is going to look for side scatter. It's looking for the blue light that bounces off the cell, just bounces, right? And you'll see that called the SSC. If you're looking at a real flow cytometry plot, you'd see that presented as SSC, right? Okay, well, that covers the scatter, the laser light bouncing off. Now, the laser light bouncing off, its job, just so you know, the job of the scatter is to actually analyze size. That's its job. It can analyze size and it can count. So the job of the scatter, these two detectors, their job is to look at size, right? So they're used, and we'll talk about what kind of data you get here in a bit. They're used to analyze how big a cell is. Now, what about that whole color thing, Reef? You did this whole demo with these cool colors. You promised us glowing cells. Yes, the cells will glow, right? So if you use labeling molecules, right, to make the cells glow, when they get hit by that laser beam, the molecules, certain of these molecules that you added to your cells, are going to absorb that blue laser light and emit another color of light in a process we call fluorescence, which I'm not going to go too much into because it's my absolute fave. I'm a huge fluorescence fan. I could talk about fluorescence for days. I can bore you with my knowledge of fluorescence. I love it. Fluorescence is the best chemical technique ever. I don't care what anybody else says. Don't listen to those organic chemists about how great NMR is, okay? I know you don't get that yet, but you will when you take chemistry classes. Fluorescence is better. Now, fluorescence is basically the glowing, if you will, right? So you're basically shining one light on something and then it glows another color. And so the fluorescence light actually radiates out from the cell in all directions, but the instrument's gonna focus on the light that goes in this upward direction, which is why I left so much space for us, right? So what's gonna happen is you're actually gonna get, most flow cytometers will look for 3 colors. The standard is 3 colors. They're gonna analyze green light that's emitted, yellow light, oh, can you see that? Yeah, you kind of can, right? Sorry about the yellow may not appear very well. And red, I apologize for that. So your 3 colors, the standard 3 colors that flow cytometers will look at are green, yellow, and red, all right? And so what they do is this light is shined, it radiates off the cell, right? So the cell is glowing with this color. So these wavelengths, these colors are gonna go in this direction. And so what they're gonna run into first is they're actually going to hit a special mirror. And so I'm gonna draw that mirror in green to remind us. So this is a special mirror. And what this mirror does is it actually bounces, reflects green light. So it's gonna bounce green light, but it lets the other wavelengths of light pass. So the red light goes right through the mirror and the yellow light goes right through the mirror. Now, the green light is then detected, as you probably guessed. We need some way to detect it. We're going to use a detector that will detect for the green light. The green light hits that detector, and the detector can see it and go, wow, that's really green, or whoa, that's not very green, right? The detector can actually tell us how much green light it sees. Very cool. And now this process is going to repeat. We're going to go to yellow next, and that actually is, by the way, strategic, because yellow is the next highest energy, in fact. So there is a reason for that. The yellow will bounce off its special mirror, and the yellow will head to its own personal detector that detects the yellow light. The red light passes through this special mirror, and the red light goes to its own special mirror that bounces the red light to yet another detector.
Dr. Reef, isn't it in decreasing energy?
Yes, it's in decreasing energy. They're in the order of decreasing energy if you followed Roy G. Biv. You'll notice the Biv is here, and we're going backwards from Roy G. Biv. The red, orange, yellow, red, orange, yellow, green, blue, purple, indigo, violet, and all that. But like, yes, it's awesome. And it's actually strategic. It's how these mirrors work. These mirrors work on energy of the light, the wavelength. And so they're typically called, these would be called long pass mirrors, where they pass the long wavelengths. Each one will bounce short wavelengths and pass longer wavelengths. And so that's why they're strategically made to bounce those colors. But yeah, that's getting more technical than I was planning on getting. But yeah, yes, it's awesome. They are strategic in the order of that bouncing. Now, okay, so we've got a cell that glows potentially, right? Depending on whether it has the molecules we put in with it.
Randy, we do have one more question I just asked.
What's another question?
How would the mirrors let some light pass?
So they have this film on them, and that film is reflective for specific colors. And so it gets really physical. And so I don't really want to go into the optics. I don't actually know what kind of materials they use. I'm not an engineer. But the idea is they have a film that is reflective for certain wavelengths of light. That's how they do it. It's really cool. If you want to study that, by the way, the physics major is for you. It's your best friend. The physics major, we've got optics tables. You can do stuff like this in physics, right? Upstairs in the labs. Very fun times. Physicists, they get very into this. This is called optics, right? And they do spectroscopy, laser spectroscopy. We actually have a laser spectroscopy lab in our physics department. Hint, hint, nudge, nudge. You know what I'm saying? It's awesome. All right. So any more questions before? Okay, I got one. There's a question. I can't see it. What is it?
Oh, it's not a question. I was just saying physics is terrible, but.
No, physics is great. It's not as good as chemistry. Chemistry's better, but physics is great. I love that. All right, so right now our picture isn't very complete, right? It looks great in theory, but we got to have something that combines all these detectors. Okay, these detectors are looking at this colorative light, right? These colors. Well, what actually looks at them? What does the analysis? Well, what do you think does the analysis? What do you think all these detectors are connected to? Can anyone guess? I'll give you a hint. You're staring at one right now.
Is it computer of some sort?
You got it. They connect to a computer. Now I'm gonna draw the oldest school computer you've ever seen, right? It's got like disk drives. Yeah, right. It's got the monitor on top, big old monitor, right? Oh yeah, that was terrible. This is my ugly computer. That's right. The point is it absolutely goes to a computer. It goes to a computer that runs special software that is able to process all of basically the detector signals all at once. And so for a single cell going through, the computer will see its intensity in red, how much yellow it's glowing, how much green it's glowing, and its scatters. And so the computer processes all of that. Yeah, I saw a really cool question. Hit me.
And they're just commenting on, they're loving your computer drawing. They're saying it's
beautiful. Oh, isn't it the best? I know. It's like really, this is like old school. This is like what I started using computers on. And then I would actually draw the keyboard here. This was me. This was me like when I was your age. Like I had this like keyboard and it was like this little disk drive and it'd go ding dong when you got on the internet. Yeah, those were me, by the way. That was my day. Right. I had dial-up when I used the phone and my mom would yell at me for using the phone. All right, I know, I went to school when dinosaurs roamed the earth. So the computer can process all this. So just to remind ourselves, size, right? And so this is measured, the size analysis, I can't spell, is measured using the ratios of our forward and side scatter. Now the cell type, so the type, like if you're doing like, am I looking at a B cell or a T cell? The cell type, you analyze using the color, right? So you using these labeled, they're actually typically labeled antibodies. Yes, they're antibodies. Antibodies are awesome. I won't get off on that rant too, because gosh, I love antibodies. Antibodies are super cool proteins, but these antibodies are typically used and they'll bind to specific cells. And so you'll get specific cells to glow, and they typically look like this. They look like this little Y, and then they put on a probe molecule. So I'll draw you one that's got like red. So they're like a red antibody, and these will go and bind to the cells, and those are actually what make it glow. You have to add a molecule, right? Pretty cool stuff. Now, okay. So that's the analyzer. Now, what does our data look like? So we said the computer can see all that at the same time, right? Now, we need to talk about the data, the third piece, right? That I would be remiss if I didn't teach you about what the data looks like. You know, I wouldn't be doing my job. So the data, right? So what it basically outputs. Data. There are actually, oh, come on, focus. I'm sorry about that, focus, okay. There are actually several types of data outputs. We're gonna focus on one today. We're gonna focus on what is very famously called the dot plot. So a dot plot is a graph of two, basically, of the components that the flow cytometer can see. And so the most classic dot plot used in flow cytometry would be a graph typically of the SSC. This is that side scatter, if you remember. So the amount of side scatter versus the amount of the forward scatter, right? And so this would be on a scale, we'll call it 10, as in 10 has lots of forward scatter, 10 means lots of side scatter. But the plot would look like something like this. And what you'll see is basically that every cell becomes a dot, right? And so every cell is a dot. So here I'm gonna draw 3 cells that are low in forward and low in side scatter. By the way, that probably means they're small cells. They're very, very small. And then we'll have a couple other cells. And so we'll see here, 1, 2, 3, 4, 5, 6. Let's do 7 because 7 is a great number. And so what we can see is we ended up with 7 cells over here, and these cells would typically be considered large. If they're high in both the SSC and the FSC, if you make your plot, and you would see these would be large cells, and these 3 would be little cells, right? So these would be your 3 small cells, right? Now, of course, a real flow cytometer is going to do this with millions of cells, as we can analyze up to 10,000 cells a second, right? So it'll be able to see a massive number of cells and then tell you how many are there, which is really cool. So this is one example of a dot plot. This is a classic dot plot, and this one is doing size. The other type of dot plot you can do is a plot that does, say, type of cell, right? Now, we talked about the colors being what we use for type. And so I'm going to make my plot with the same general scaling right here. So 10, 5. And what we're going to graph this time, and I hope my marker works, is we're going to graph the red intensity here, so how red the cell is on the y-axis. And we're going to graph the yellow intensity. I know this is hard to see, but it's actually more real, right? So real life, they love to compare yellow and red. The yellow intensity, right? And so what you'll get is you'll get a dot plot, and I'm going to draw a couple cells here. We're going to look at 10 cells again, and we're going to see 3 cells. I'm going to say 3 cells that are actually low in both, meaning they're not red and they're not really yellow, right? So they'll appear here, low on the y-axis and low on the x-axis. Then I'm going to draw 2 cells, I hope you can see it, that are nice and yellow here. Notice that they are high on the yellow scale, but low on the red scale. Then I'm going to draw 3 red cells, cells that are high on the red scale and low on the yellow scale. And then just for total funsies, because it's awesome, if you had a cell that was both red and yellow, well, right, I'll draw them as orange. You can actually have cells that have both, right? And so this could be some special type of cell. And I'll draw them as orange for us. I hope you can see the color. I don't know how well the color comes through, but isn't that fun? And so you could see, we could actually do a percentage analysis here. You can see that I've got 10 cells. And so you can see, well, I've got 30% of my cells are this red. So maybe this is a B cell. I've got 20% of my cells are yellow. Maybe that's a T cell. So you can analyze type by doing that. Does that make sense? Okay. Is that a question or a yes?
It's a yes.
Excellent. Now, this is where when I give this lesson to my poor Chem 254 students in Chemical Analysis 2, I love this lesson. They may not. It's at the very end of the semester. I love throwing it out there. This is a very bioanalytical technique. So this is very classically biology meets analytical chemistry. It's blood analysis, it's cell analysis, right? So it's a little bit of a niche technique for a chemist, but I teach my students it anyway because it's awesome. Now, if I were teaching them and you were in my class, if you were here today, the next thing I would give you is I would pass out one of these. And this is my assignment. This is a representative assignment that I give my students, where I give them like what real dot plots look like, right? These are some examples of what real dot plots will look like. And I give them a task. And if you were here today, which I'm so sad that you're not, we're not here hanging out, but if you were, I would send you home with a baggie of M&Ms. And your job would be to count your M&Ms and be the flow cytometer and determine what you've got. Is your bag of M&Ms, you pretend it's a sample of blood from a patient, and you can identify whether they've got physics disease, biology disease, or chemistry degrees. That's obviously a joke. Those aren't real diseases. Those are jokes. But it's really fun, right? And so you would basically count yourself the number of M&Ms and call them cells and say, well, do you have more than 40% of them expressing a certain protein? So are they, like, say, red? So you'll see that it looks for red for protein X and yellow for protein Y. And then if an orange cell comes along, then it's got both proteins X and Y. And so, boy, if it's orange, man, that's chemistry disease. That's the worst. Just kidding. Chemistry's the best. Okay. So you'd be walking out with these. I'm so sorry you're not. It's a bummer. But I would be handing those over to you. I wish you were here in person with me. It'd be a lot of fun. We'd have done this on the whiteboard. It'd have been really cool. All right. How does that sound? Does that sound pretty good? All right, I'm going to go ahead and
wonderful.
I'm going to go ahead and stop sharing now. So now that is the end of the lesson. Now I'll admit in my real class when I teach that, I'll teach that with actual detector names. I'll expect them to know the names of those mirrors, that they're long pass or dichroic. There are actually multiple types you can use. But I can get more technical about why we use what we use. Why do we use that 488 laser? That's actually, the blue laser is a 488 nanometer laser. We talk about that, typically an argon ion laser. We get all kinds, we can get much deeper into the why we do what we do from a chemistry standpoint. But we didn't care about that today. Today is just how awesome it is. All right. Oh, okay. Bye, Amanda. That sounds good. Head now. But so now, okay, so we've got a couple minutes left. I know you all have to go. You all are going to be headed to the awesome administrations panel where they're going to talk about, I'm sure, all kinds of awesomely cool stuff. But I am a faculty member. I'm in chemistry. I know all the biologists. I know the physicists. I know the program, I would say, decently well. So I wanted to open it up now for questions from you. And I can actually, now that I'm in this view, I could actually, I guess, look at, oh yeah, I can look at the chat now. So I can see your faces and the chat. This is exciting. So does anybody have any questions? Anything they want to know? How many students do we get that are on the pharmacy path? We probably, I'll admit, as a liberal arts institution, we don't get a ton of them, although they absolutely are here. Most students that choose that path oftentimes come in. I see them come into basically the chemistry major or the biochemistry major would be my two favorite for that path, by the way. I recommend biochemistry or chemistry. I would say we get probably 3 to 5 a year that end up deciding to go on the pharmacy path. I like that. Okay, wow, this is cool. Will these recorded seminars be posted somewhere? Kelly, that's you. I don't know.
Yep. Yeah, I put yes. They will be posted on our YouTube channel, and so hopefully within the next week or so, we'll get all of these up.
Oh boy, that's terrifying. Good luck. Have fun with that. Now I'm gonna be all over the website. Cool. I'm gonna be famous. Awesome. Okay. Great. How does the college grading system compare to the one in high school? Honestly, I don't actually really know. Classically, I can speak from basically how I would grade. So depending on the class you take with me, college in general, if I'm talking generalities, college classes in general put a lot of focus on a few exams. Now, if you talk to someone like me, I like having more of those exams. So like my general chemistry class would have 5 exams. You will hear the stories though about some college classes, and some will have like 1 midterm and 1 final, and then all these other assignments around it. And so it puts this massive amount of stress on those 2 exams. That is still done, but most faculty at UMW don't do that. That is something that generally, as a faculty member, if I were talking to my colleagues, I would say to my colleagues, why do you do 2 exams? You're going to stress out the students. Like, that's kind of ridiculous. If they fail one, how do they pass your class? I don't like that model personally. So I do a multiple exam model to help basically balance out the grading system. So you'd have multiple exams, you do quizzes with me. I'm a big fan of quizzes that are lower stakes, so we can see what you know and don't know. I do homework in general chemistry. There's an online homework. So there's a lot of other grades. Then of course there are labs that come into your grade. So there's a lot of other grades. So students can be poor exam takers and still pass my class. I've got so many other grades. There are other things they can do really well on and get through my class, no problem, get a good grade. So I hope that kind of answers the college grading system compared to high school. I don't know much about the high school grading system. How would biology minors work next to a major in environmental sciences? It'd be fine. Environmental science, one of the great things about environmental science is that it's kind of a combination of many of the different basically majors. You're taking a lot of topics from chemistry and biology and physics, and you're merging them into basically one science that focuses on the environment. And so it's very easy to do a biology minor with environmental sciences. A lot of them, basically just have those classes, some of them will overlap. And then you'll take additional classes in biology to make that minor work. Yeah, that's very common. How would I say, oh, you're very welcome, Samantha. How would I say the average bio or chem class is structured in general? Oh, I gave that, okay. So basically I gave that. So I would do, generally we do multiple exams, typically 3 exams on a final. I do 4 exams on a final in my gen chem class. I know that biology would do 3 exams typically, I think, in a final. They'll typically have an online homework. Then they'll have the lab reports. So you've got a lot of grades going in. That's pretty standard. And some professors will do participation, some won't. It kind of depends. A lot of them do quizzes, some don't. But I know you'll have basically a set of exams, you'll have labs, you'll have homework in the chemistry and the biology classes. Heavy workload, it kind of depends. Would I say that the classes have heavy workloads? Yes, I'm not gonna lie to you. Science classes are 4 credits because of the lab components. You have an extra 3 hours of lab a week. You're basically in lab for 3 hours a week, plus the 1 hour every 3 days or an hour and a half every 2 days. depending on what class you sign up for. And so, yes, there is a fair amount of workload in the sciences. I'm not going to lie. I won't sugarcoat it. Yeah, and chemistry is notoriously challenging. It's not as bad as everyone makes it out to be. I think it's fabulous, right? It's not that bad, but the sciences are a fair amount of work. I won't lie. Yeah, the expectation of work is pretty high. What courses would you say are the best to take for anyone interested in forensics? Forensics. If you want to do forensics, I highly recommend doing forensics. I would argue for a chemistry major, a pure chemistry major, actually, because it gets the analytical classes, which is, basically classes with techniques like flow cytometry, or they do things like GCMS, basically gas chromatography, mass spec. Forensics utilizes instrumentation in chemistry, and we teach that in our chemistry curriculum. So if I were doing forensics, I would go chemistry, and then I would take biology classes that interested me, like the cell type biology classes, because knowledge of cells and understanding of cells and how they work and DNA will help you with your forensics work later. So I would then add biology to it. It's pretty fun. You could do it as a biochemist if you wanted to. If you really like the biology, you could do a biochemistry degree and then add the analytical classes to get your forensics. Does anyone do psych and bio together? Yes, people have done psych and bio together. I won't lie. It's a challenging course load in terms of the number of courses. So because bio and psych, they have some overlap, mainly in the neurosciences, basically. So that we have a neuroscience minor, which is very popular and very cool, by the way. I love it. And that combines basically the neuropsychology classes with the neurobiology and cell biology classes. We have that minor that works really well. But yes, people will do both. It's a pretty challenging course load. You want to get your gen eds taken as fast as you can. Basically, if you were doing that so that you could focus your final years on the psych classes and the bio classes that you need to graduate. It's pretty intense, but I have absolutely seen students do psychology and biology together. Yes. Any advice for students wanting to work towards a science major? Wanting to work toward a science major. So what is, So is that like someone who doesn't, so Samantha, is that question like someone who doesn't know what kind of science they want to do? Is it like that kind of? I mean, like, if you don't know, what I would probably do is when you come in, when you first start in your first year, I would try and take, the science classes, the intro science classes that most interest you. So let's say you really liked, you were like, I loved biology in high school and I liked the environment. So maybe you try to take biology 121 and the earth and environmental science like 110, right? And so you take their intro course. And so like you could do both of those intro courses and see which one you like better. And then in the second semester or the second year, you can start honing in on those classes. But one of the nice things about all of the sciences as a whole is that the first year courses, they gel really well all together. So a lot of times we'll see science majors, like when you major in one of the sciences, you probably will have taken at least one or two of the other science programs intro courses. So biology, for instance, requires general chemistry. So if you're a biology major, you will have taken general chemistry at a minimum. Earth and environmental science, I think, also requires chemistry because it builds on that. I'm pretty sure it also uses chemistry in its degree. I know one of their paths actually has like upper division chemistries that you can take that count towards your major, which is pretty cool. So I would argue that all of the sciences are pretty linked. And so you kind of just start on what sciences you like the best and go from there and take more classes in those. Any other questions?
Oh, so I was wondering about like virtual classes. Did that make like learning chemistry, bio harder? Because I'm not sure if we might go back during the fall. I hope we do though.
So virtual classes, I'm working under the assumption that we are going to be back very largely in person. I'm going to follow what the provost has gone on record as saying, 90% in-person classes. I am excited for in-person classes. I think the students are more successful in in-person classes in general. The average students are more successful in in-person classes. I am definitely a better instructor in person. I do my best with this. I really do, but I'm sad for all of you because you're not getting the best me. The best of me is seeing me where I can like jump around in front of you and make ridiculous gestures and fall over, and like I'm much more entertaining when I get to move in person when I'm at the whiteboard. So like I'm sad. I think I'm better at it in person. I mean, I know I'm better in person. So I'm bummed that you all only got half of me through Zoom instead of the all me. But that's a great question. Virtual classes though are taught much like this. There are actually lots of different styles for the sciences and how they teach them. So I don't know. It really depends on your faculty member how they would teach the virtual class. There are several different styles. But if you like the idea of face-to-face, I highly recommend you take one of the classes that would be listed as face-to-face, so that you know you're going to be going into the classroom and that professor is going to be there to teach you. If you like the idea of virtual, some people do, you could take the virtual, you could pick the virtual section.
All right, thank you.
No problem. I hope that answered your question. How do you believe COVID impact experiments? So you mean like experiments like lab work in the fall semester? Is that what that how do you believe COVID will impact experiments? So COVID, we've done actually, I would argue, yeah, no, I mean, we've done a really good job at UMW. I am super proud of our departments. We've done a fantastic job at implementing in-person labs. The in-person labs right now, we run half as many students, right? So we have the specific rooms with their specific sizes. And our students are, I would argue, you may be doing slightly fewer labs in person. A lot of times the labs now are worked on half of the classes in person and the other half is doing a virtual assignment at home where you're doing some kind of data analysis. What's really awesome about it though, is that it has changed fundamentally how the experiments you're doing when you're in person, you're doing them. There's no like lab partner team up. I would argue that my students just did in a lab this week 7 titrations where they had to do it on their own. And so it was a fantastic experience for them. Like they were like, at first they were like, this is so hard. And then they were like, well, I practiced. And they, got some tips from me and they were like crushing it. They're like, wow, I got really good at this. I'm like, I know you got good at this. Of course you did. You're practicing. Like you're crushing this thing. You can do this. And so I would argue that actually you get a great experience doing experiments on your own. You just don't do quite as many experiments. So it's a trade-off. You do more experiments when we're in person, when everybody's back and COVID's not around. But yeah, and my research, I still do research. My research students can still work. Like we have research students in the lab. I still work with students. We use the microscope and my students grow cells. They work on microfluidic devices. I have an undergraduate research program. I teach lots of students and they still do research. Favorite class to teach? That's a tough one. I would actually argue biochemistry. I know it sounds weird. I'm betraying my PhD. My PhD is in analytical chemistry. I'm betraying it by saying that. I have fallen in love with teaching biochemistry. Biochemistry is taught to juniors and seniors, kind of as a mixed class, depending on when you want to take it. And it's got a mixture of biology majors and biochemistry majors and chemistry majors in the class. It makes the class so much fun because all the students come with a different perspective and they ask different questions because they're coming from different background knowledges, and then they learn from each other. And they bounce ideas off of each other and they're like, oh, you learned that in cell bio? Maybe I'll go take cell bio to learn that. Like as a chemist, it's awesome. It's amazing. All right, yeah, the admin panel is getting ready. Yo, everybody should get going. You don't have to listen to me talk. This was awesome.
I can post the link here for everybody.
Thank you so much.
Yay. Oh, you're so welcome. This was lots of fun.
It was nice.
I'm good. I'm glad you think so. That's great. I wish I could give you the M&M bag for homework. Stupid COVID.
It's okay.
Shucks.
One day we'll get the M&M bag.
One day you get the M&M bag. That's right. One day, the M&M bag.
Oh.
Bye, everybody.